Have you ever thought about wanting to fix a genetic problem or design your own baby? Well, you're in luck. With the new advancement in genetic altering called CRISPR, you can change it all. Just go to www.crispybaby.org.
CRISPR has been around since the 1960s, where scientists bombarded plants with radiation to cause random mutations in genetic code. The idea was to get useful plant variation by pure chance.
In the 1970s, scientists inserted DNA snippets into bacteria, animals, and plants to study and modify them for research. And in 1974, the first modified mouse was born, making mice the standard tool for research in saving millions of lives.
And in the 1960s, the first plant was given a microbe to absorb excess oil. In the 90s, scientists treated infertility by making embryos that had 3 parents or 3 different sets of chromosomes. And then, in the 1994, the first artificially made food was the Flavor Saver tomato was put on the shelf.
You can give a copy of your DNA you want modified and put it into a living cell with Cas9. CRISPR acts like a GPS system and offers the ability to edit live cells, switch the genes on and off, and target and study particular DNA sequences. It's so easy to do it yourself. All you need is the lab.
The Cas9 protein is acting as a molecular scissor, cutting DNA in locations in humans, microorganisms, and animals. CRISPR stands for clustered, regularly interspaced, short palindromic repeats, and their mission is to develop transformative gene-based medicines that offer curative treatments.
Treatments include ex vivo and in vivo. Ex vivo treatment is where the patient's cells that are bad are removed from the body. Then the Cas9 is delivered to the cells in culture, resulting in the desired edit. Finally, the therapeutically modified cells are expanded in culture and returned to the patient. This treatment is for homeoglobinopathies, such as sickle cell disease, and CAR T cell therapy for any type of cancer.
In vivo treatment is when the Cas9 therapeutic is packaged and delivered to a target organ such as the liver. They are delivered through nanoparticles.
Going back to the beginning of the video, CRISPR in the future will be much more advanced. Surprise babies will be a thing of the past. In the near future, anyone with an excess amount of money can plan their own designer babies. Designer clothes are not included. Parents will be able to decide features, smarts, and athletics.
On a more serious note, Cas9 has the ability to change humanity forever. They are working on ending all genetic and non-genetic diseases and working on even eternal youth.
Though many people are on board with genome editing, others are not. One problem is that large genomes contain multiple DNA sequences that are identical or highly homologous to intended targets. CRISPR could also cut these sequences, causing cell death or transformation. Another big risk of germline editing therapy is that the introduction of it has unforeseen side effects that would be recognized only in generations to come.
Also concerned that we will head on a slippery slope to using editing for non-therapeutic and enhancement purposes. While scientists have been experimenting in labs, many animals have been put at risk, making animals become close to extinction.